Hey everybody, you haven't met me before. My name is Armin Swift. I am the program director and lead instructor for Biomanufacturing program here at OSU OKC. And welcome. I hope we get along real well. My plan is I'm going to have a really good time teaching these courses. You, who sadly are going to be stuck with me, I hope you will join me in having fun with this, because biomanufacturing really is, to me at least, super exciting, quite a great career choice, and it's fun. It's just, I mean, it's a hoot, right? I love it. So anyway, that's me. You and I will be seeing each other quite often, either virtually or in the classroom. So I hope we get along, and I hope you'll join me in having a really good time at this. Some of the coursework is certainly challenging. This is biomanufacturing, but we can get through it, and it's my job here to help you guys succeed. So let's keep it at that. So what are we here to talk about today? Well, it's pretty short and sweet. I'm going to talk real quick about what is biomanufacturing. So biomanufacturing is where we make a product, but in order to make that product, we really have to do it in a biological system like a bacteria or a yeast, or in some cases, mammalian cells in culture. It's all pretty cool. Well, especially mammalian cell culture. That's super cool. You all will learn that at a later date. But some of these products, many of them therapeutic in nature, drugs or proteins, like for instance insulin. I'm sure you've heard of insulin. That's actually considered the first biotechnological product or biomanufactured product back in 1972, I believe it was. Before that, all insulin that was used for diabetics was derived from the pancreases of either pigs or cows. And while it was quite similar, it was certainly mammalian. It was quite similar to human insulin. It was not an exact match. I can't talk today. And so this new product, human insulin, derived from human genes inserted into E. coli or Escherichia coli, produced the first real biomanufactured product, which was human insulin. Super cool to look up the history of that. And I will probably talk about it in some of our live classes as well. But biomanufacturing at the core is we're manufacturing a product, generally an enzyme or more likely a protein, and we're doing it inside of a living system like a bacteria, a yeast, or mammalian cells, into which we have inserted a gene or genes of interest. In the case of insulin, we had two different vats of E. coli. Into the E. coli in one vat, we inserted the gene for insulin precursor A, because there are two precursors to insulin in the human system. And in the other vat, in the E. coli in that vat, we inserted insulin precursor B. We let those grow out in huge vats. We took each one of those vats and we mushed everything up, and we purified out the insulin precursor A and the insulin precursor B. And then we chemically combined them into human insulin, sweet, pure human insulin, not porcine nor bovine. So that was a really great thing. And now we have lots and lots of other products. I mean, lots and lots of other products, therapeutics from monoclonal antibodies that are made in something called a hybridoma that we'll talk about later, and other proteins, things like that. That's really basically it for this one. I'm going to sign off now. Please be sure to watch the 2 or I think 3 videos after this one. They will explain things a lot better. Thanks so much, and I'll see you later.